Hello everyone. So we are going to talk about the recombinant DNA technology. So basically we are going to discuss on what are the enzymes used in the recombinant DNA technology. So firstly we are discussing about the history. Stanley Cohen and Herbert Boyer produced the first genetic altered organisms in 1973. They are the scientists who produced a first successfully recombinant two plasmids PS C101 and PSC102 and cloned in the new plasmid in EcoRI. So now we are going to talk about RDT. So RDT is the generation of DNA fragments and selection of the desired piece of DNA. एग्जाम्पल ह्यूमन जीन. सो हम RDT में क्या करते हैं कि हम एक डिज़ायरल अपना सीक्वेंस लेते हैं और उसे इंसर्ट करते हैं एक प्लाज्मिड के अंदर ताकि हमें एक रिकमेंडेंट DNA मिल सके. So then we inserted of the plus selected DNA into a cloning vector, example plasmid, to create a recombinant DNA or chimeric DNA. So introduction of the recombinant vectors into host cell, जैसे कि bacteria, multiplication of and selection of clone containing the recombinant molecules. Then the expression of the gene to produce the desired product. So basically here is our donor DNA and this is our plasmid DNA. So we can use the restriction endonuclease to cut that, or we get the desired DNA piece and we get the desired plasmid DNA. So we further then ligase that and we form a recombinant DNA sequence, recombinant DNA. Then we introduce into the host cells E. coli. Then further we multiplication of selection of the clones. Then these are the our recombinant DNA implement in the E. coli. So this is a protein encoded by cloning gene. So this is the basic principle of recombinant DNA technology. So we are going to discuss enzymes that are involved in the RDT. So first one is the DNA ligase. DNA ligase is isolated from E. coli and bacteriophage commercially and used in the RDT. The enzyme DNA ligase join the fragments with cloning vector. Now we are going to discuss about reverse transcriptase. Reverse transcriptase is used to synthesize complementary strand cDNA from mRNA template, or it is also known as RNA dependent RNA polymerase. It is isolated from retroviruses. Then restriction endonucleases. Restriction endonucleases recognize and cut DNA strand at specific sequence called restriction site. सो जो रेस्ट्रिक्शन एंडोन्यूक्लियस होता है वो ना रिकॉग्नाइज करता है और कट करता है डीएनए के स्ट्रैंड को फ्रॉम अ स्पेसिफिक सीक्वेंस कॉल्ड रेस्ट्रिक्शन साइट्स. These enzyme is isolated from wide variety of microorganisms. Endonucleases enzyme degrades foreign genome when enter inside microbial cell, but the host cell own DNA is protected from the endonucleases by methylation of base at restriction site. There are three type of restriction endonucleases. Type 1 restriction endonucleases is it has both methylation and endonucleases activity. It require ATP to cut the DNA. It cut DNA about 1000 base pair away from the restriction site. It has example of EcoKI. Then the type 2 restriction endonucleases. It does require ATP to cut DNA. It cut DNA at a restriction site itself. Example EcoR1 and Hinge3. Type 3 restriction endonucleases. It require ATP to cut DNA. It cut DNA about 25 base pair away from the restriction site. And example EcoPI. Fourth one is the terminal transferase. It is an enzyme that converts blunt end of the DNA fragment into sticky end. If the restriction endonucleases cut DNA forming blunt end, then the efficiency of ligation is very low. So the enzyme terminate transferase convert blunt end into the sticky end. So जो टर्मिनल ट्रांसफेरेज होता है वो क्या करता है कि अगर कोई स्टिकी ब्लंट एंड प्रोड्यूस हो गया है, कट हो गया है, तो वो उसकी एफिशिएंसी बहुत लो हो जाती है, तो वो कन्वर्ट करता है ब्लंट एंड को स्टिकी एंड में. Terminal transferase enzyme synthesize short sequence of complementary nucleotide at free end of DNA so that blunt end is converted into sticky end. Then the further is nucleases. The enzyme nuclease hydrolyze the phosphate diester bond on the DNA strand, creating 3OH group and 5 phosphate group. It usually cut DNA on either side of distortion caused by the thymine dimer or intercalating agent. The gap is filled by the DNA polymerase and strand is joined by the DNA ligase. Nucleases are two types, endonucleases and exonucleases. So the next one is the DNA polymerase. DNA polymerase is a complex enzyme which synthesizes nucleotide complementary to template strand. Then it adds nucleotide to the free 3OH end and help to elongate of strands. It also help to fill gap in the double-stranded DNA. DNA polymerase 1 isolated from the E. coli is commonly used in the gene cloning. Tag polymerase isolated from the thermus equitis is used in the PCR. Tag polymerase. Then the ribonucleases H, RNase H. RNase H remove mRNA from DNA RNA heteroduplex, and that mRNA is used to synthesize cDNA. It is isolated from the retroviruses. Then the alkaline phosphatase. The enzyme alkaline phosphatase help in the removal of terminal phosphate group from 5' end. It prevents self-annealing of vector DNA soon after cut the open by restriction endonucleases. Then the last one is our polynucleotide kinase. It adds phosphate group from ATP molecule to terminate 5' end dephosphorylation by alkaline phosphatase. So that's all about the enzyme used in the RDT. So hope you like it.